Hello everyone, welcome back to my channel, The Biolab Scholar. So in my previous video, I talked about how you can retrieve a gene sequence by three different ways and my personal favorite, phytosome. In this video, I will tell you about how you can design the primers and how to name the primer for a better organization that will help you at the later stages of your cloning. So in general, primers have certain characteristics, like there should not be any secondary structures or hairpin structures in a primer. There should not be primer dimers forming between the forward and the reverse primer, and the GC content of the primer should be between 40 to 60%, and the difference in the annealing temperature of forward and reverse primer should not be more than 5%. So these are the general characteristics, but you are going to see certain exceptions when you are designing primers for cloning. So yes, again, depending on your experiments, there are different softwares available to design your primers. So in this video, I am particularly telling you about how you design the primers for cloning. So let's get started. So for this, I'm going to share my screen again so that you can see how I am doing, how I'm designing the primers. So yeah, yes, now you can see my screen. So here I have already taken a gene from phytosome, and you can see beautiful color coding. I literally love it. And I have named the gene ACT1, but it's not ACT1 gene. I'm just taking the example. So here you can see the green region is the 5' UTR, the blue one. Here you can see ATG where the first exon starts, and then the white portion is the intron. The light blue region is your second exon, and then the purple region is your 3' UTR. So how to design a primer? So before designing a primer, I want to tell you how we actually read the sequence. That will help you to understand how I name the primers when I talk about it later in the video. So ATG is basically the starting codon of a gene. So in this ATG, A is marked as number 1. So the following nucleotides are in chronological order. So T will be 2, G will be 3, A will be 4, this A will be 5, and so on until we cover all the 3' UTR region. The region upstream to this A, like which is the 5' UTR region, is marked as -1. So we'll read -1, C is -1, T is -2, A is -3, T is -4, and so on until we have the 5' UTR region. Or maybe even if we go in the promoter, I'll talk about the promoter thing maybe late at the later stages. So this gene is like, I have taken a very small gene so that it is easier for you guys to understand how I am designing the primers and how, and you can use the same formula for any gene, maybe big or small, it doesn't matter. So let's get started with designing the forward and reverse primer. So designing the forward primer is much easier than the reverse primer. So for the forward primer, what we do is we directly copy our a little bit of sequence from the ATG site, starting from the ATG site, and then we go up to maybe 24, 25, maybe 25 to 30 days where it's good. So I'll just count the word. Okay, it's 22, and maybe I'll take 2 or 3 more. So this region I feel is good. So this is 26 nucleotides now. I'll copy this region, and now I'm going to paste it here because I'm going to use it later for naming the primer. And for this primer, I also want to check if there are any secondary or secondary structures or primer dimers. I'll copy this. How to check the secondary structures and primer dimers is by using the oligocalc. So how you open your oligocalc, I have already opened. So I'll show you how you can open your oligocalc. You just type oligocalc, and then you see this option, oligocalc, oligonucleotide properties calculator. You just open this option, and then you just paste your primers, and then go and calculate, check the self-complementarity. So we'll go, the other window will pop up, and then here it shows there is a potential hairpin formation. So if I talk about the normal primers, this is not a good option, but then I said you will see certain exceptions. So this is one of them. Even if you see certain hairpin or primer dimer formation for your cloning primer, there are chances that they are going to amplify, because we are using different kind of tag and tag polymerase and conditions to amplify the gene for cloning. So there are chances that this primer is still going to work. So it is better that first you try to amplify your gene with this primer, because this is the best primer you can have. It's from the beginning of your gene, and there will be like no extra sequences from the 5' UTR. So first try using this primer to amplify your gene, and then if for some reasons you cannot amplify your gene, what you can do is you can take the 5' UTR region to design your second primer. Yes, you can go in your 5' UTR region and design another primer. So here, for example, I'll take another primer from this to this. I'll just count the word count. Okay, this is good. This is 25 base pairs. I'll just copy it. And then I'll again go in the OligoCalc. I'm going to check the self-complementarity, and then this primer is great. It doesn't show any hairpin, any primer dimer, so it's really good primer. So you can use this primer, but I would still suggest you to use your primer from the ATG first. The primers still work, like it has worked for me even after showing some bit of primer dimer and hairpin structures. It has amplified my genes. So it is a good idea to amplify, to try amplifying, and then if it doesn't work, then go for the second option. So I'll also paste the second primer, so that at later stages I will tell you how the naming of the primer differs if you take it from the exonic regions, and if you take it from the 5' UTR region. So for the reverse primer, it is a little bit tricky, because for the reverse primer, you have to make the reverse complementary sequence of your primer. So if I take a primer this long, I'll just check the word size. Okay, this is perfect. This is 24 base pairs. And I will go in the reverse complementary software. Here you can just search on the Google reverse complementary, and then this option from the bioinformatics.org. You can open this option, and then they have shown like your sample sequence, but then you can just delete it. You can put your own sample, own sequence, and then submit it. So another pop-up will open, and then you have the reverse primer. So I'll just copy it and then paste it. So I'll just copy my sequence. So this is my actual reverse primer. Then I'll also mark on my sequence the region that I have used for making my reverse primer. So I'll just go and highlight it. Yeah, so that I can track later when I'm going to show how we name the primers. So let's get started with naming the primers. So this is my forward primer. How we name our primer is we keep our the name of our organism, that is AT, which is Arabidopsis thaliana, and then the name of the gene, ACT1, and then at what nucleotide you have started your forward primer. So in this case, because we have taken the ATG, so A is number 1 position. So here we will write 1 and then forward. So this is our name of the forward primer. So it is important to keep the name of the organism. It is the name of the organism is optional, because if in your lab you only have just one model organism, you can completely eliminate, because there is no possibility of having any other organism. But like in my lab, I have multiple organisms in my lab. So it is important for our lab to keep the names of the organisms so that it is more organized. So name of the gene is very important. The position of the nucleotide that is going to be a part of your insert is very crucial, because this, you need to know a precise nucleotides of your insert. These things are going to help you at the later stages of your cloning, that I will discuss in the next step, next video, why this is important to keep the position of the nucleotides. And then whether it is a forward primer or a reverse primer. How we name the primers from the 5' UTR is same as the above. So I'll write ACT1. It's just the nucleotide position will change. So because we have taken this portion, and the upstream region is in the minus position, so we'll just go and check how long we have taken. So because I have started from here, I'll just mark it and then count until I reaches 80G. And then I'll see what is the word count. I'll just check. So it is minus 27. So I'll write as minus 27 forward. So you can see a difference in the naming of both the primers. So this was your position 1. This is your minus 27 position. This minus 27 position itself shows that your primer is from the 5' UTR, which is very important to know that what is the precise nucleotides in your insert that I already mentioned. For your reverse primer, so the initial things are same. It's just the you have to include how long your complete insert will be. So if you have taken, let's see, if you are only designing the forward primer from 80G, then you will select from 80G to all the way until you reaches the stop codon and then word count. So this is your 401 bases long region. So you are going to write 401 RC. And you might now have a question like, what if I have designed my primer from the 5' UTR? It doesn't matter, because the counting will still be the same. You are counting in positives from 80G only. You do not count from like A to all the way until stop codon, and then you do the word count and you write like 428. No, this is wrong. You only have to always count from your starting site, like from your 80G to your reverse primer. Even if you have like, for example, if your this reverse primer doesn't work, then you have to design a primer from your 3' UTR region. So how you will count that is, I'll just highlight, and then how you will count is from 80G to all the way in your 3' UTR region, and then just word count. Review and then word count. So 433 will be your position of your reverse primer. So just take care that in your forward primers, you are going to mention your first nucleotide, where is your starting site. And in the reverse primer, you are going to mention the last nucleotide of your primer. You cannot like count until here and then say that, okay, this is the position of my reverse primer. No, this will be wrong, because this region of your reverse primer is going to be there in your insert. So you need to know like the whole thing your insert is. So it is recommended and it is essential that you count from the last nucleotide of your reverse primer. So this is your actual counting. So yes, that's it for today's video. I hope you find the video helpful. If you have any questions, you can comment down below and I'll try to answer them. And in my next video, I'm going to tell you how you include the enzyme sites, and then you are going to know why knowing your precise insert is important. So yeah, stay tuned. See you in the next video. Till then, take care, stay healthy, stay happy. Bye bye.